Okay, so we want to look at this is where we stopped. So we want to continue the part two. So we want to look at what an immunogen is. And an immunogen is anything that elicits an immune response. So we have macromolecules, generally either proteins or polysaccharides. Proteins are most potent immunogens. Why? Because when you use proteins as vaccines, they are able to elicit immune response, which will end up helping you to get a very good secondary response or memory. Lipids and nucleic acids do not serve as a good immunogen. So when you use them as vaccine, you will not get any protection. For cell-mediated immunity, only proteins and some lipids and glycoproteins serve as immunogens. Okay, so we use only proteins and some glycoproteins. Proteins must be presented together with major histocompatibility. I will teach you this one later. Lipids and glycolipids that can elicit cell-mediated immunity must also be combined with MAC-like membrane molecules called cluster of differentiation 1, CD1. Okay, so these are some of the antigens or vaccines or organisms which are dead or weakened or attenuated inside some vaccines, and they are able to elicit immune response due to their foreignness, that they are foreign to the body. If they are the same as the cells of the body will not fight it. It's like somebody giving you blood group A and you are also group A. Your body will not react to it. But if you have blood group A and they give you blood group B, your body produces antibodies against group B. So there will be what? Agglutination reactions and serious consequences. So chemical composition, ability to process and present whatever will come there. Okay, so lipids as antigens. Lipids are used as haptens and attached to suitable carrier molecules such as proteins, keyhole limpet, hemocyanin, or bovine serum albumin. So let's see, what is a hapten? A hapten is just a molecule. These are haptens. They are just molecules which, when they are carried by a carrier molecule, they become a full-blown or very strong or potent antigen which is able to elicit immune response. So hapten in itself alone cannot elicit any immune response. Likewise, carrier molecule, which is like fat or lipids, cannot elicit immune response unless the two of them are joined together before they can be dangerous elicitors of what? Immunity. They can potently produce immune responses. So when you put this combination or this conjugate, this combined complex, that's hapten and carrier complex, this one into a human being or somebody, it will elicit strong immune response, and so it serves as a vaccine. By immunizing with these lipid protein conjugates, it is possible to obtain antibodies that are highly specific for the target lipids. T cells can recognize lipoidal compounds such as glycolipids and some phospholipids via MAC. I will teach the major histocompatibility. Okay, so adjuvants. Let's look at adjuvants. They are substances that, when mixed with an antigen and injected with it, enhance the immunogenicity of that antigen. Like I said immunogenicity is what? The ability of an antigen to elicit immune response. Well, there are some, so if you inject water into my body or somebody's body, there will not be any immune reaction. But if you inject an organism like malaria, there will be reaction. That is immunogenicity of the malaria parasite. So that's how it is. So that is immunogenicity of the malaria parasite. Okay, so let's see. Antigen adjuvants are often used to boost immune response when an antigen has low immunogenicity. So when the antigen has low immunogenicity, like the polysaccharide vaccine. The proteins are better than what the glucose vaccines. So if you have a vaccine containing an animal that is a microorganism that is a protein, it means that that vaccine can protect you for about 10 years and over. But if it is glucose and others, the maximum of 3 years. So when only small amounts of an antigen are available, so if you don't have much antigens available, the antigen is not able to elicit a good immune response like the carbohydrates or the glycoproteins one, like the carbohydrates immunogens or antigens. Then you have to add what an adjuvant to strengthen or to increase the immunological response. So adjuvants function by what it prolongs the antigen persistence. Okay, so you can read on this one. So comparison of antigen recognition by T cells and B cells. Okay, so you read on these two. So vaccines. This one, vaccine, we've talked about vaccine. So let's see. So vaccines are, we have the live, okay, so the fundamental principle of vaccination is to administer a killed or weakened is the same as attenuated form of an infectious agent or a component of a microbe which does not cause disease but elicits an immune response that provides protection against infection by the live pathogen. So you give a vaccine that is harmless, then when the harmful organism comes, your body can clear it. So we have different types of vaccines. So vaccines are most effective if the infectious agent does not establish latency, that it doesn't hide in the genome. If the organism establishes latency, it means that if your cells are like this, and your nucleus is like this, and your DNA is like this, the organism can come and incorporate this DNA inside your genome. So as your DNA, so this is the gene. So as your cell divides into two daughter cells, your DNA will also divide into two daughter cells. Your DNA will also divide into two daughter cells. As your DNA also divides into two daughter cells, the HIV virus is also dividing. So this is what we call latency. It does not interfere, so a vaccine are most effective if the infectious agent does not interfere with the host immune response. And if the infectious agents are limited to human hosts and do not have an animal reservoir. So some of these infectious agents, they are found in humans, but they are also found in animals, and the animals don't become sick. It's the humans that become. So if you touch a goat or you eat a goat meat and it has HIV, you end up getting it. But the goat itself, the HIV cannot kill it or have an effect in him. So that is what we call organisms that can, viruses that can attack animal hosts. We call it what? Zoonosis. Zoonosis. Infection in animal. Zoonosis. That can be transferred to humans. We call zoonosis. Okay, so look at antibodies, attenuated and inactivated bacterial and viral vaccines, first generation vaccines. So go and read on all these. Go and read on them. Prefer antigens. We've done a lot of vaccines. So go and read on this. Some of them will appear in the exams. So go and read. Okay. Thank you very much. We will now meet. Our next class will be on. Our next class is going to be on hypersensitivity reactions, then the major histocompatibility, which I'll try and merge the two. so that you pick it as one slot. So without much ado, have a good evening and a good night. Sleep well, dream about Jesus, pray hard, love God, and He will direct your path. Thank you.